Alrighty. Okay. Hello, everybody. My name is Megan Motley. I am a PhD candidate in Douglas Menke's lab at the University of Georgia. And today I'm going to tell you guys about RPL6Y as a candidate for primary sex determination in Anolis segrii, the brown anole, and kind of walk you guys through how we've identified this gene and why we think it is the most exciting candidate for primary sex determination. So across the animal kingdom, we see striking morphological differences between males and females of the same species. And these can be differences in color, shape, size, plumage, appendage, structure, any of the above. And often these animals are using these for mate selection, preference, fighting, you kind of name it. But underlying these sex-specific morphological differences is an organism's sex determination system. So what is sex determination? Well, sex determination in most vertebrates is when the bipotential or undifferentiated gonad goes on to develop into either an ovary and produces eggs or a testis to produce sperm. And so this sort of starts with a primary sex determination initiation cue that then can initiate the downstream pathway of gonad differentiation. And sex determination is absolutely fundamental to species survival and reproductive success. And the timing and accuracy of this is obviously very fundamental. If we're not producing offspring, our species will not survive. And while we know a lot about how this works in mammals, we know, a bit about how it works in birds, our understanding of the genetic mechanisms underlying this process in reptiles is really, really lacking. So in my lab, we use the brown anole, which is a part of the Anolis species group. There are over 400 species of Anolis lizards, and they drastically differ in size, shape, appendage, and habitat, as you'll see here in my tree diagram. We've got lizards with long limbs that live at the tops of trees, and we've got lizards with short limbs that live in bushes and things like that. And so Anolis has become a really excellent group to study evolution, ecology, comparative genomics, climate change biology, and in the Menke lab, we use it mostly to study limbs and eyes. And so I'm sure you guys are wondering why I'm here talking about sex determination. Well, the Menke lab has done a ton of work to establish Anolis segrii as a model species, including lots and lots of genomic and genetic resources, functional genetic studies. The brown anole in particular is invasive. It's very easy to keep in the lab, and we have an XX reference genome, and we've been able to do CRISPR gene editing in the brown anole. So while I told you guys that Anolis looks, ooh, my goodness. While Anolis looks incredibly different across species, sorry, it's still kind of going crazy. I am not touching any buttons. So while Anolis looks incredibly different across species, all Anolis, all 400-something species, they all share an XXXY sex determination system. And this sex determination system was derived, my goodness, was derived from a single ancestral autosomal pair. I'm afraid every time I talk, it's going to change. So this single ancestral autosomal pair, this XXXY system, arose what we believe to be around 160 million years ago, and is conserved across anoles, and then it's also conserved across iguanas, and essentially almost all pleurodonts share this XXXY system. And so we hypothesize that wow, it is just trying to, maybe. I will also say that everything on this computer is in French, and I do not speak French, so I'm also not sure what buttons I'm supposed to be pushing. Alrighty. essentially we hypothesize that whatever gene is driving sex determination in Anolis segrii should be the same gene driving male sex determination in all Anolis and iguanas, pleurodont lizards that are using the same sex determination system. So when you go to identify a primary sex determination gene, like I decided to set out to do in 2019, it's really helpful to have a Y chromosome to use. We didn't have one. And so that was sort of the first step. What are we looking for when we're looking for a sex determination gene? Well, based on what we know in mammals, which is again another very ancient XXXY system, we're looking for a gene that we thought would be located in the most ancient region of the Y chromosome, expressed early in XY gonads, right? We need gene expression in our early embryonic male gonads to be driving this, and then we're hoping to see it conserved across pleurodonts. And then additionally, if it is still an XY pair, the Y gene, we're expecting to have a decent amount of divergence from its X pair. So again, you need a Y chromosome. So when I got to grad school in 2019, all we had was our XX reference genome and the 2011 XX genome of a green anole. Neither of those are very helpful when you're trying to find a Y sex determination gene. So what did we do? I went out to Florida and I actually caught this lizard here with my hands, and we sequenced its genome. So we did PacBio hi-fi sequencing, and then we did high-C sequencing and scaffolding. Phase Genomics did that for us, and what we got was a really good Y genome. So we got 30x coverage across the genome, 15x coverage for the X and the Y, and our genome is actually, the size of the genome that we got suggests that both the alleles have been captured for most of our genome. Working in anoles, they are highly polymorphic, very, very outbred and wild. We literally are working with lizards that we've caught from Florida and brought back to Georgia. So what we found, unsurprisingly, is that the Y chromosome has undergone massive gene loss. So in the X-specific region, we still have around 1,100 genes. In the Y-specific region, we only have about 340 genes. And then I don't have time to show you, but in the Geneva paper from 2022, our brown anole XX genome, we were able to identify sort of the oldest region of the X, which we have then now, sort of identified the oldest region of the Y in our Y chromosome. So then what did we do? Well, I have done more RNA-seq than any grad student could hope, want, or need to do. But I wanted to look at gene expression across developmental time, right, in these gonads. And so what you're looking at here is this big pink tissue is the mesonephros, sort of the precursor to the kidney. It's a very large piece of tissue. And then on top of that tissue, as you'll see in sort of this white color, are our embryonic gonads. So for my stages 7, 8, and 9, I actually took those gonads off of the mesonephros and ran RNA-seq libraries based on some pooled either XX or XY samples. And then for stage 5, because the gonadal ridge is really just starting to form, the way that I've done that experiment is I just sort of left it on the mesonephros and kind of cut off any of the extra mesonephros. Unfortunately, we didn't get a ton of signal from the gonad for my stage 5 libraries, but the mesonephros libraries, I made libraries that didn't have the gonad, and those libraries have served as a really good control. So again, what are we looking for? We're looking for genes that are going to be expressed in early XY gonads, conserved across pleurodonts, and then if it's an XY pair, has the Y diverge significantly. So what we found is there are only 5 genes left in the ancient Y region. And out of those 5 genes, most of them are not good options for sex determination. So CCDC74B was not conserved in iguana, so that kind of doesn't check the box, right? We're looking for something that would be conserved across any species that has this XY sex chromosome. RSPH14 was exciting. It's been known to work in spermatogenesis, but when we looked at our transcriptome data, there was absolutely no expression in early XY gonads. So again, not useful for what we were looking for. And then GNAS and UBE2L3, while they are exciting because they lack sequence divergence, UBE2L3 is one of the most conserved genes we've ever seen. Which is exciting, but again, not what we're looking for in terms of a sex determination gene. So it left us with the gene RPL6Y. And so what is RPL6Y? Well, it's ribosomal protein 6. Again, not what you would normally find if you're looking for a sex determination gene. But what we saw is that it was expressed in early XY gonads, and that RPL6Y has only about 70% sequence identity to RPL6X. And when we did some preliminary searching from either available pleurodont genomes or publicly available pleurodont genomes or genomes from anoles that have been sequenced in collaborations that we're in, we found RPL6X and Y present in 11 pleurodonts. So what does it look like? Alrighty, so RPL6Y in XY samples. So we've got our normalized read counts as a proxy for level of expression. And along the bottom, you'll see stage 5 is mesonephros, stage 7 forelimb. So again, these are going to be our non-gonadal tissues. And then we've got our stage 7, 8, and 9 gonad in our male samples. And so as you'll see, stage 5 mesonephros and stage 7 forelimb have very low expression. And what we're seeing here, right, is a very high peak at stage 7 and then sort of tapering off. I don't have expression data from stage 6, but I believe it would be more highly expressed at stage 6 than even stage 7 from some of my work looking through the stage 7 data. We're a little bit too far in sex determination. So great, but what does RPL6X look like? So RPL6X, what we actually found was in our mesonephros and our forelimb samples. So in light pink and light blue, that's going to be our non-gonadal samples, either in our XX gonads or our XY gonads. And then again, we've got our stage 7, 8, and 9 gonads. Dark pink and dark blue are either XX or XY gonads. So what you'll see pretty drastically is that in non-gonadal tissues, RPL6X is very, very highly expressed. And then when we actually get into our gonad samples, we're seeing very low expression relative of RPL6X. And so we went back to our genome and kind of just wanted to look and see what the actual gene structure looks like. And what we found is that RPL6X in blue on top sits really closely to this gene PTPN11. And in our non-gonadal tissues, right, we've got our regular PTPN11 transcription, our RPL6X transcription, all looks normal. But then this is what we saw in our gonad samples. And we were like, okay, what is going on? This is not what we would expect. And so what we actually found based on these patterns is that the gene PTPN11 has a gonad-specific promoter that actually is in front of the RPL6 gene. And based on these splicing patterns, we're actually seeing transcription of an antisense transcript for RPL6X and anolis, and we're going to call that Astra. So what does Astra expression look like? Again, we've got our mesonephros and our forelimb. You'll see that the expression for Astra is 0. It's not being produced, as you saw in the splicing patterns. But what we see in stage 7, 8, and 9 gonads is obviously very different. So we're getting high expression of Astra. What does this look like kind of as a whole glimpse into XY expression? Again, we've got our stage 5 mesonephros, our stage 7 forelimb, and then we've got our stage 7 XY gonad. And so in the darkest blue, that's Astra. So again, it's at 0 and it starts to tick up in our gonad. In the next shade of blue, we've got our RPL6Y expression, again, turning on in that gonad. And then in the lightest blue, we're going to have that RPL6X expression that's extremely high in our non-gonadal tissues and then lowered in our gonadal tissues. So what is this? RPL6X, RPL6Y, and Astra, a novel mechanism for sex determination. So in XX embryonic gonads, Astra and XY embryonic gonads, Astra expression is being turned on. RPL6X expression is being decreased. And then in our male gonads, RPL6Y is turning on. And that's leading to either ovary development if you don't have RPL6Y, testis development if you have RPL6Y. And so what does this mean, right? Ribosomal proteins, I know I was not expecting to be thrown into the world of ribosomes, but what we're seeing from some some new research coming out, so in this first paper, male germ cell specific ribosome controls male fertility, they actually knocked out a male specific ribosomal protein and saw reduced male fertility. And then in the second paper here, ribosome heterogeneity in Drosophila, they found that ribosomal populations in testis and ovary were the most heterogeneous populations compared to the other tissues they had looked at. So what we're seeing is like mounting evidence that sex-specific ribosomal proteins are being specialized to regulate translation. So what we don't know is Astra blocking transcription, degrading post-transcriptionally? We don't know. But what we do see, right, is that Astra is turning on in gonads, RPL6X is being lowered, and RPL6Y is then turning on. And we believe that RPL6Y is a testis-specific ribosomal protein that is driving male sex determination. I don't think this is unique to anoles. I think we have a very narrow way of looking for sex determination genes, and I think this kind of can blow up the field, right? Now, for 5 years, we've been overlooking this RPL6Y gene. It was reported in 2017 in the Green and All paper. And throughout my grad school career, we've been like, oh, it's a ribosomal protein. We don't care about that. But actually, from our, out of the 5 genes left, it's really the only option we have. So what are my future directions? Going to continue analyzing pleurodont genomes. We're going to be utilizing this CRISPR gene editing to functionally validate RPL6Y as the initiator of primary sex determination. We've actually already started doing this, so please come talk to me. I don't have enough time to tell you about the surgeries we've been doing. And then my last, hopefully, experiment in the lab will be using mRNA and C2 to determine the locality of expression for RPL6Y and Astra. Sorry I went over. Thanks for bearing with my weird slides. Thank you, and I'll take any questions if we have time.
All right, hopefully it works this time. Oh, hello everyone. My name is Adrian Lee. I'm originally from Taiwan, but I'm currently working at the University of Florida with Dr. Gavin Naylor. Welcome to my talk about the mysterious sex chromosomal shocks and rays. So we know that almost all vertebrate species have 2 sexes, but triggers for these sex determinations can be pretty diverse. Generally speaking, these can be put into 2 categories, the genetic sex determination and environmental sex determination. In genetic sex determinations, the triggers for these sex determinations are genetic components. These can sometimes be the entire chromosomes. These can be heterozygous in male, known as the XY system, and heterozygous in female, known as the ZW system. While in environmental sex determinations, the triggers for these sex determinations are environmental cues. These are usually temperature, but there are sometimes other factors. And the majority of our understanding in vertebrate sex determinations come from the tetrapods. We know that, for example, most mammals have XY system, while the birds and snakes mostly have ZW, and some certain groups of tetrapods, such as sea turtles and crocodilians, have temperature sex determination. While in bony fish, the sex determinations can be pretty complicated. If they are having GSD, they often can have multiple sex chromosomes or have their chromosomes, sex chromosomes morphologically identical. And that leads us to the shocks and rays. So the reason why we want to study the sex determinations of sharks and rays is because they actually belong to a group of vertebrates known as the chondrichthyans that has diverged from the rest of vertebrates a long time ago. However, they have a much lower diversity compared to the sister group. So this is part of our effort to study the biology and to try to understand how these organisms can survive for such a long time with such a low diversity. Also, because of their position on the phylogenetic tree, if we understand their system, they can help us to infer the ancestral state of all jawed vertebrates. So one of the most common ways to study the sex determination is by observing their karyotypes. Much like the example I'm showing here, the human male karyotype, this method is great at detecting sex determinations where there are differentiated sex chromosomes, but it will fail to detect homomorphic sex chromosomes or temperature sex determination systems. In the terms of sharks and rays, out of the 13 orders, we have only 3 orders we have any species reported as having differentiated sex chromosomes, with 2 more having potentially unreliable reports, while the rest of it, we do not have any reports of differentiated sex chromosomes yet. Out of these 3 orders, we have 2 orders having some species reported as having an X and Y chromosomes, while in the stingray order, the myelobatiform is being much more complicated. We have some species showing XY, some species having the second pair of the X chromosome, the X1 and X2, and one species even reported as having the XXXO system, where the Y chromosome is completely degenerated. So another way to investigate the sex determination is through their genome assemblies. So recently, there are more and more reference genomes of sharks and rays being assembled, many of which were done by the Vertebrate Genome Project, which has generated a lot of high-quality reference genome, and some of which have their gene annotated, some of them even have sex chromosomes identified. So the objectives of my project were to use these available genome assemblies to identify and validate the sex chromosomal pairs, and to investigate the genetic components on these sex chromosomes, and lastly, to conduct a chromosome synteny conservation analysis to try to figure out how these chromosomes evolve over time. And here are my results. The first part of my result will be focusing on our genome and short read mapping. And the first example we have is the white shark species, whose reference genome is estimated to be 4.3 gigabase pair and a chromosome count of 41 pairs. And this reference genome actually have their X and Y chromosomes identified. And the method we apply to validate the sex chromosome identification is called the chromosome quotient method, which is by sequencing the short reads of females and males of the same species and then map them against the reference genome and calculate the female to male coverage ratio across each chromosome. In terms of the X and Y system, we should expect the ratio on the autosome to be 1:1, and on X chromosome to be 2:1, on Y chromosome to be 0:1. Like for example, if you look at this graph of the mapping result on an autosome, what we see is that the female to male coverage ratio, which is on the Y-axis, across the entire range of the chromosome, showing on average a 1:1 ratio. If you look at the X chromosome, what we see is that almost the entire range of the X chromosome, we have the closely expected the 2:1 ratio female to male coverage, except for the last 2 million base pair, which having a 1:1 ratio. So this indicates that there's the range of the pseudo-autosomal regions, which is a region that are still recombining, but nonetheless attached to the rest of the sex chromosomal group, pairs. We see the similar region on the Y chromosome, as well as the majority of the region showing the closely expected 0:1 female to male coverage ratio, except for the first 2 million base pair, which again having the 1:1 ratio, indicating a PAR. So another example we have here is the epilay shark genome, who also have their X and Y chromosome identified. So on their X chromosome, we see again almost the entire range. We have a 2:1 female to male coverage ratio, as expected, except for the first 300,000 base pair 1:1 ratio, indicating potential PAR. However, this region, the PAR region, is much more difficult to identify from the Y chromosome. The last example of this method is our Thorny-Skate reference genome, who has a chromosome count of 49 pairs and only the X chromosome identified so far. However, this identified X chromosome also seems to be a little bit problematic, since our mapping results show that its female to male ratio across the entire chromosome having been 1:1, similar to what we see on the autosome. Upon digging down a little bit more, we realize that another chromosome annotated as chromosome 46 actually have this expected of the X chromosome, the 2:1 female to male coverage ratio. We will therefore suggest to appoint this chromosome as the actual X chromosome of the species. As a summary of this past part, the white shark and epilay shark genome, we using a chromosome quotient method validated the X and Y chromosome status, and we also identified potential range of their PARs. While in the Thorny-Skate, we would like to appoint the chromosome 46 as their actual X chromosome of the species. The next part of my result will be focusing on our synteny analysis. So our synteny analysis, which is based on 1:1 ortholog analysis, is basically taking the genes on the chromosome of one species and see where the orthologs are on the other species. So for example, let's take a look at this graph between the white shark on the top and the epilay shark on the bottom. We see that the majority of the genes that we found, so these are 1:1 orthologs, found to be on the X chromosome of the white shark genome is also found to be on the X chromosome of the epilay shark genome. If we zoom in onto those genes, what we realize is that almost 50% of the genes that were found to be on the X chromosome of either the white shark or the epilay shark were actually found to be on the X chromosome of the other species. So this is quite an incredible case of conservation because these two species are not necessarily closely related as they diverged sometime back in the Jurassic. So as we keep comparing more and more species pairs, we realize this conservation of X chromosomes are actually pretty widespread, and these include the distantly related species pairs, such as white shark with sawfish, with devil ray, and thorny skate. So another thing that we realize from these analyses is that the chromosome X1 and X2, which are unique among the two species found in the Myliobati forms, seem to have different origins. So it seems like chromosome X1 could be inherited from the ancestral X chromosome that is shared among sharks and ray species due to how many genes they shared with all the X chromosomes of all our species. While the chromosome X2, it seems that they were repurposed from an autosomal pair sometime after they split up from the rhinopristiforms happened in the Triassic. Yet another application we realized from this analysis is that due to after we realize how conserved these X chromosomes can be for sharks and ray species, we can use the ortholog analysis to try to infer where the X chromosomes are in reference genomes where they haven't been identified. For example, we have the reference genomes of white-spotted bamboo shark and little skate, neither of which have their X or Y chromosomes labeled yet, but after we map the X-linked genes of the epaulet shark and thorny skate, we realize the most likely candidate for the X chromosome in the two species are chromosome 43 in the bamboo shark and chromosome 40 for the little skate. Summary of this past part, the 1-to-1 ortholog analysis showed that X chromosome genes are conserved even among distantly related species, and this conserved synteny of the X chromosome can help us to infer where the X chromosomes are in any reference genome of sharks and rays to come. And the last part of my result will be focusing on our brief characterization of their sex chromosomes. So from our synteny conservation analysis, we noticed that there are 23 1-to-1 ortholog groups that were found to be on the X chromosomes of every reference genome of sharks and rays available. So this potentially indicates that there was a pair of ancestral chromosomes that gave rises to the sex chromosomes of all of these shark and ray species. So outside of these very conserved regions, there are at least 1,200 genes that were found to be on the X chromosome of at least one species of sharks and rays. So if we map these results onto a phylogenetic tree, this is what it will look like. So the ancestral pair that gave rises to all the X chromosomes can be more than 300 million years old, while each lineage is sharing more and more genes on their X chromosomes, and also like certain species within the Myliobati forms have their unique X2 chromosome. So another question we have been asking is that what are the genes that were found to be on the sex chromosome of sharks and rays? And the first sets of genes that we look into were those genes that have been shown to play an important role in the sex determination of some other vertebrate species, such as SOX3 and DMRT1. However, none of these genes were found to be on the sex chromosomes of sharks and rays. Instead, what we repeatedly found to be on the sex chromosomes of these sharks and rays are genes such as HUC clusters and ZBTB39. So to our knowledge, none of these genes have been directly linked to the sex determinations of any vertebrates yet. So it seems to us that last branch likely have some kind of genetic sex determination system whose mechanisms we do not know from the vertebrates yet. So here are some take-home information. So sharks and rays have a long evolutionary history, and evidence suggests that sex chromosomes came from one ancestral chromosomal pair. And outside of these, at least 23 genes of XCR, the conserved regions, X chromosomes can be pretty diverse. So some of them have a different length of PAR, and some of them can have a second pair of X chromosome. And although we do not know what their master sex-determining genes can be, it seems that their mechanism is something different from what we know from the vertebrates so far. As our ongoing and future works, we are currently acquiring more high-quality genome assembly, as well as getting short-read data, and also the RNA-seq data for gene function analysis. And lastly, we're also trying to collect the karyotyping data for the independent verification of these sex chromosomes, as well as for fluorescent in situ hybridization. And with that, thank you very much for your attention. I'll be happy to take any questions.
Good morning, everyone. My name is Lydia. Things are also moving really quickly here, so we'll see what happens. I'm a PhD candidate at UBC, and today I'm going to present a little bit of my PhD work. I think we're in a bit of a time crunch, so I'm going to get right into it. I'm going to get us all started on the same page with sex chromosome evolution and some brief theory behind how they evolve. So the canonical theory states that they evolved from a pair of autosomes, so we have these little yellow chromosomes, and one will acquire then a sex-determining gene, indicated by the blue bar. Over time, you can get recombination suppression around that sex-determining gene, which can result in reduced efficacy in selection. You're no longer able to break up linkage between deleterious mutations. And that can then lead us to divergence between the chromosomes and eventual degeneration, like you see in chimpanzees and humans, where our male Y is relatively small to the X. Of course, we also have a lot of evidence that sex chromosome evolution is not linear with time. And you can get all sorts of things happening. So you can get a loss of the Y chromosome in the spiny rats, for example. You can get persistence of the chromosome size, so no degeneration. You can also get turnover events, where its sex chromosome essentially hops to another autosome and restarting that cycle. And so there's a lot of things going on. And so a lot of my work focuses on the guppy, and I'll kind of tell you why they're so great. Its scientific name, Pussylia reticulata, has been a model system to study adaptation, sexual selection, and also an inspiration for sex chromosome evolution. So we have a little female depicted by Jocelyn Shu with no colors, and you get these beautiful charismatic males with different colors, different patterns. But that's not the only reason why we study them. So one of the reasons why they're great for sex chromosome evolution is that they have homomorphic sex chromosomes, meaning that the Y is relatively the same size as the X. And also they're fairly young relative to other sex chromosomes, only being about 20 million years old. So here at the very top, I have Pussylia reticulata highlighted, sex chromosome 12 in that middle panel, and you can see it's actually shared with some of its closest relatives. So you can see the orange dot, that's the origin of their sex chromosome. And also on the very right panel, you'll see its sex chromosome karyotype with the females in red and blue are the males, and just another emphasis on Y, that homomorphic sex chromosome. And so going to keep you kind of on the hook as to why this is so fascinating. A little bit of work has been done in the lab already from a former postdoc, Pedro, who found some evidence of a small region of divergence between the X and the Y. So here labeled as stratum or a sex-linked region. He had gone out to the wild, collected a bunch of guppies, and compared the male to female genome. And he found the sex chromosome depicted on the X-axis reduced male to female coverage in a region of the stratum. So on the Y is that normalized coverage. You can see at the one is mostly at the ones, showing equal coverage between males and females, and at the very distal end, there's that purple box, the stratum, where you see that dip in the male coverage. And he also found further evidence looking at male-specific single nucleotide polymorphisms, or SNPs, which are just variants in the genome, and showing that there's, in that same respective region, an accumulation of the male-specific SNPs. So yeah. To kind of bring us back to where we were, this is the image that I just had earlier. And we have circled the guppy, why they're so fascinating. We have evidence of divergence, but not very much has been done past that. So I mentioned degeneration. We don't see that in the terms of the size of the sex chromosomes. But what makes them a great opportunity to study sex chromosome evolution is that we can have an opportunity to look at degeneration, see if it's actually occurring and how fast it's occurring, and what sort of form. So degeneration can also be a loss of gene function or gene expression, which is kind of what my work is focusing on. And so I looked at the male-specific SNPs that Pedro had also found, but I'm also looking across not just the sex chromosome, but seeing if it's present also in the rest of the genome. So in order to do that, I used the same data set, so 60 males, 60 females, and I followed the same protocol to call SNPs and filtered them. But I also use a program called SNPF, which is able to predict the effect of the SNP on protein coding sequences using a reference genome from Ensemble. So just to let everyone know, we have a female reference from Ensemble. And with being able to predict the SNP effect, you can actually try to call what sort of impact it will have on the genome. So I looked at deleterious SNPs, which I labeled as either a SNP causing a stop gain or stop loss, or a missense mutation. And as a reminder, missense mutations are changes in the nucleotide sequence that actually change the amino acid produced and also the phenotype of the protein. Okay, jumping into some results. So I looked at the deleterious SNPs again, this time on the sex chromosome. So again, X is the sex chromosome. And this time on the Y-axis, I have a male to female SNP count. So anything that you see above a 0 is a male-specific deleterious SNP. Anything below that would be female. And you have a little legend on the side there to show just how many individuals had a SNP present there. So the more blue the dot is, the more individuals. All right, we have the stratum labeled again in purple, and you can see all these little blue dots that are present there, and one little blue dot that's not, or a little bit outside of the stratum. So we get that accumulation of deleterious male-specific SNPs, which is what we expected if degeneration is occurring. We're seeing that there's an accumulation of potential impact on these genes, especially in the stratum. Of course, is this a unique phenomenon on the sex chromosome? We're not sure. We had to look at the rest of the genome to find out. So here I have essentially the same sort of plot with chromosome positions and size on the X. The Y will be showing now all the chromosomes of the genome, but the plot will be the same. You can see the another legend in red is now the female-specific deleterious SNPs. But I'd like to emphasize again that the female doesn't have anything unique in its genome. The male is the one that has the Y. Females have 2X. So anything that is a female-specific SNP is just a false positive. It's kind of how we're detecting how well our program is working. All right, big plot. So there's all the chromosomes. You can kind of see a few dots here and there. We have 27 male-specific deleterious SNPs and only 14 female-specific deleterious SNPs on the autosomes, about half of what we expect to see. And to kind of focus your eyes back on that sex chromosome, we're actually seeing about 2.5 times more deleterious SNPs relative to the rest of the genome. So again, this is really suggesting to us that degeneration is occurring through deleterious impacts on this region of divergence in the sex chromosome. But to kind of go a little bit further, we want to know whether these genes are still functional. So in order to do that, we looked at expression data, but this is still really ongoing work, so this is very, very preliminary. In fact, I'm only going to showcase 2 genes that we currently have data for. So these are genes that had deleterious SNPs in them that were male-specific. So just throwing the names out there. We did find a couple other genes. Don't have time to talk about them today, but you can come find me afterwards and we can talk. So we looked at Y expression of these genes, so very specifically at the Y. And in order to do that, I used single-cell expression data that was collected from another former postdoc, Yulia. And this time it was collected from 15 males and 15 females of the lab population. And in order to look at expression, I used a proxy of allele counting and comparing the minor allele to the major allele. So equal expression would then be a ratio of 1 between the 2 alleles. And if we see a ratio of less than 1, it's a reduced minor allele expression. All right, some preliminary results. I'd like, again, very much to emphasize that a lot of this data is still being processed, so very, very preliminary. But here I have the gonad tissue present with the 2 genes that I mentioned that were here on the x-axis. And on that y-axis, again, is that ratio of minor to major allele, where that one line represents equal expression. What does it look like? So the red is the female gonadal tissue, blue is male gonadal tissue, and you can see at that the red tissue is all sitting at that one line. So there's equal expression between the major and minor alleles, but the blue actually is quite reduced relative to the red. So we're seeing a reduced minor expression allele relative to the major, and in the case of males, this minor is the Y allele. So what we're really seeing is a reduced Y allele expression relative to the X, or less expression on the Y chromosome relative, or that at least the stratum relative to the X. Again, this is great. This is kind of what we were expecting if degeneration is occurring. We're seeing a loss of gene expression. Of course, again, I said very preliminary, so when all the data has been processed, and a lot of the power that comes from single-cell data is actually to be able to pull out the cell types. So once all the data is processed, I'm going to take the tissue and dissect it into the different types of cells, and I'll be able to compare that to somatic tissue to see if this pattern holds. All right, so some conclusions. I've already mentioned that there's been evidence of region of divergence in the X and Y of sex chromosomes in Bacillia reticulata, which is great. It's a great starting point in understanding what's going on in the system. But we wanted to also know if degeneration is occurring and how fast it's occurring and what form it takes. So I do find evidence of early degeneration through accumulation of deleterious SNPs, especially in the stratum. But I also have very preliminary results that suggest that the impacted genes have reduced expression on the Y relative to the X. Ongoing work, so stay tuned as more data gets processed. I'm happy to talk about that. All right, thank you for that talk. I think we're back on time, so if you guys have questions, feel free to ask. I think we have time for a question or two. Yeah, Keith.
Do you have evidence that the deleterious SNPs are associated or causing the gene, the reduced
I guess if not super sure, so the genes that I have pulled up for the reduced expressions are the ones that do contain the the deleterious SNPs. And so what I'm hoping to do once I have all that data is to be able to sparse out the haploid cells from the gonad tissue to really focus in on the Y. That kind of. Oh yeah, I actually haven't looked at that yet, so yeah, I can definitely do that though. Any question? Sorry? Yeah, so we actually don't know where what the sex-determining gene is in the guppy yet. So this is definitely the candidate region to try to find the sex-determining gene. And so actually one of the great things about it is that we can look at these deleterious SNPs, see where they're occurring, what sort of genes are involved, and then yeah, I can look into the sex-determining gene. So, one more question?
So, do you find any dosage compensation in this trial except for undosed genes that
Yeah.
degenerate?
Yeah, so actually, I can maybe go back. I don't have this in the slide, but so the region isn't super diverged from each other, so we do have like a few of those genes. And so we aren't necessarily expecting to see like complete dosage compensation, but you see there's some species that are highlighted in the red branches that do have like a very degenerated Y chromosome, and we do see dosage compensation in that. So we do have like an idea of maybe where dosage compensation might occur in this phylogeny, but yeah, not necessarily here in the species that's not super, that doesn't have a super diverged chromosome. All right, I think that's my time, so.
Okay. I didn't put mine as a PowerPoint, so I don't know how to like present this. There we go. That's good enough for me, honestly. If anyone else has an issue with that, I'm really sorry. Okay, hang on, let me just. All right, so hi everyone. My name's Cleo. I am a PhD candidate in Dr. Anthony Geneva's lab at Rutgers Camden, and today I'm going to be building on all of the fabulous talks that came before me to talk about my research, which is about investigating neosex chromosome evolution in a novel XXY lizard genome. So as most of the previous speakers already said, we've seen that sex chromosomes have this outsized impact on evolution compared to autosomes because of their hemizygosity. So there's only one copy of each chromosome. And processes like Muller's ratchet, the fast X effect, mean that X and Y chromosomes experience different evolutionary pressures, but this can actually be difficult to observe because most sex chromosomes are evolutionarily stable over time. However, a great way to observe this phenomenon of sex chromosome evolution is in species with neosex chromosomes because we can really kind of see evolutionary processes unfolding as new regions of chromosomes get kind of combined together and form neosex chromosomes. And as Megan mentioned, we love to study anolis lizards because their parallel radiation makes them excellent model for studying these evolutionary questions. And in fact, karyotype data actually highlights that anolis chromosomes have undergone fusions and fissions. So on this phylogenetic tree, we can see I've highlighted there have been fusions and fissions in this chromosomes that lead to XXY chromosome systems. However, as Megan also mentioned, sex chromosomes in anoles are highly conserved, so we should expect to see a lot of the same genes over time in these different species. And I specifically highlighted, my model system is somewhere in there. It's actually the one with the little asterisk next to it, but I study Anolis discus, and that one has a multiple sex chromosome system that resulted from fusions with microautosomes. So here is the karyotype of him. And this X1, X2Y system came about from an autosomal fusion event. So throughout the talk, I'll use the same colors for all of the sex chromosomes. So I'm going to use green for autosomes, and then I'll have X1 be pink and Y be blue, and then I'll have X2 be purple. But basically, what you're seeing here is this is what it would have looked like at the beginning before the sex chromosome fusion happened. So we have all of these chromosomes kind of lining up on the metaphase plate. And what happened is that there was this autosomal fusion of the Y chromosome that's created this neo-Y chromosome. So the autosome and the Y kind of got glommed on together there. And now we have the the former autosome now becomes a sex chromosome. It becomes our X2 chromosome, and it segregates like a sex chromosome. So that means that females will have a karyotype of X1, X2, X1, X2, and then males will have a karyotype of X1, X2, and our neo-Y, which is now really longer. So overall, my goal is to leverage our high-quality Anolis disticus assembly to identify these neosex chromosomes to better understand their evolutionary pressures. So to do this, I assembled the genome of Anolis disticus, which is the hispaniolan barcanol, and I did this by using PacBio reads, and I used Omni-High-C reads for the scaffolding, and that resulted in a very high-quality, complete, and contiguous genome. So we have actually over 95% busco completeness, so we are seeing 95% of the expected genes, and our N50 is pretty large, thanks in part to our long PacBio reads, which are very long without compromising on read quality. And so we have our assembly, so we wanted to identify our sex chromosomes. Unfortunately, we didn't have any genome expression data from this project, so we just had our assembly. So we were going to just look through our assembly to find sex chromosomes or scaffolds that we think have a lower coverage. So sex chromosomes should have lower coverage because we have a male, so we should only have one copy of the X, one copy of the second X, and the Y. And we overall found that scaffolds 8, 12, 13, and 16 had lower coverage. So these are the scaffolds that we found that had lower coverage, so we think that they are most likely pretty interesting and most likely to be sex chromosomes. Next, we looked for sex-linked genes present on the hypothesized sex chromosomes. So there are 296 known X genes, and we were able to identify them either on scaffold 8 or scaffold 12, so 87.1% of these known X genes were on either of those scaffolds. So that supports 8 and 12 as identified as the X and the neo-X, as well as 5 of the 7 known Y genes were identified on scaffold 8 or scaffold 13. So that supports scaffold 13 as part of being the Y because it's going to line up with the X. So we see kind of homology there. We also did cross-species synteny with other assemblies. So we used our Anolis disticus assembly. My advisors recently published Anolis sagrii genome and the Anolis carolinensis genome from 2011, and we found that the Anolis carolinensis X is syntenic to Anolis sagrii 7, which is also in turn syntenic to 8 in Anolis disticus. And I want to mention that we did all of the comparisons with Anolis carolinensis because that one is the one that is hypothesized to hold the ancestral X with no recombinations, or excuse me, with no fusions or fissions in anoles. I don't know why it's not clicking. There we go. Oh, are we coming in too hot? I think we are. Okay. I think everybody this morning is just having this problem, so that's really fun. This is a Satsuma synteny result mapping Anolis disticus to itself. So we mapped them to itself so that we could see how these chromosomes were interacting with each other. And we found that Anolis disticus 8 and 12 were both syntenic to scaffolds 13 and scaffold 16. However, there was no synteny between scaffold 8 and scaffold 12, which is what we expect because, like I said earlier, if they're kind of lining up on the metaphase plate like that, we don't expect 8 and 12 to be kind of interacting at all. We kind of just expect them to be like vertical. Oops. And so through all of this kind of integrative set of analyses, we were able to identify all of the sex-associated scaffolds in our assembly. So scaffold 8, which is pink, is going to be syntenic to the X chromosome in A carolinensis, and we believe that that one's the ancestral X. Scaffold 12 is the neo-X, which is syntenic to chromosome 12 in A carolinensis. And then we have our Y scaffolds as scaffolds 13 and 16. We do believe that the Y is in one piece. We just believe that we didn't fully assemble the Y because of, all of the repetitive elements that are in Y chromosomes that make them so fun and difficult to assemble, as I'm sure Megan knows. So now that we've identified all of the sex chromosomes, we're going to see whether these chromosomes' evolutionary patterns mirror what is expected by theory. I would love to talk about theory. I did a ton of reading over spring break and through the semester, but I don't have time to get into all the theory, so you're gonna just have to trust me on this one. We're going to expect the Y chromosome to degenerate. we are going to expect our neo-X chromosome to accumulate deleterious variants, and then we are going to expect our neo-X specifically to undergo purifying selection. So we're going to look through and see if we can find evidence of this happening in our genome. And the way that we're going to do that is we did that by going across the genome in sliding windows and counting up different attributes. So first we found that the Y chromosome is experiencing degeneration as measured by gene density. These are some violin plots. If you don't like to read violin plots, I'm really sorry. I don't like to read them either. But they are typically, I like to look at them as like kind of a distribution. And then I put the mean in the middle so you can kind of see where the mean is supposed to be. And so what we can see is that the Y chromosome has much lower gene density than the rest of the chromosomes, so we do expect that it's going to be undergoing this degeneration. And then next, we also looked at transposable elements, which typically have negative effects because they're inserting themselves into the genome, typically in a deleterious way. And we're going to use that as a proxy for degeneration. And so what we found here is that we found that the X2 has regions of the genome with extremely high transposable element density. So we find that the X2 is accumulating mutations, which is what we expect, and we expect that it's accumulating mutations kind of unevenly because there are certain regions of the genome that are they're more hospitable to transposable element accumulation, so that is really what we see. We have this very long tail. I also use SNP-F. So SNP-F is a model that classified each single nucleotide polymorphism according to its predicted impact. So a high impact, for example, would be something like a frameshift mutation or a start-stop mutation. And then a moderate mutation would be a substitution. And then a low polymorphism would be like something like a silent mutation where there's not really effect that we see in the genome. And then a modifier is anything that's like not hitting a coding region. But for our research, we wanted to look specifically for the moderate and high mutations. And so we found that the X2 had an intermediate density of moderate and high mutations. So overall, the density of these mutations is lower than that of autosomes, but it's still higher than both the ancient X and the Y. So that suggests that this X2 is undergoing the process of purging deleterious mutations and undergoing this purifying selection that we would see after it's accumulated all of these mutations. Unfortunately, there were some limitations to this research, as with all research. So the first limitation is that there currently was no available reptile Y chromosome. When Megan publishes her paper, we'll have one, which is really exciting. When we also have a research which is it's only a proxy of natural selection, so these are models that are only estimating deleteriousness. We're not actually going to be able to test empirically for anything. We're just going to, have a proxy of this natural selection here. However, overall, Anolis disticus sex chromosomes did mirror what was expected by theory. So we do expect the Y degeneration, and we did see that through our lower gene density in the Y chromosome. We expected to see deleterious variant accumulation, which we did, with the X2 having very high transposable element density. And lastly, we have the purifying selection. So we did find that X2 has this intermediate density of deleterious variants, which does explain possibly that it could be undergoing purifying selection. And our genome does set up future work in sex chromosome evolution, such as Megan's work, which identifies the sex-determining region in Anolis lizards, which we obviously do want to find out about. We also want to use existing gene expression data in the future to understand which genes are experiencing dosage compensation, because dosage compensation can also impact the rate at which sex chromosomes are evolving and diverging from each other. And lastly, we do want to add to comparative studies of novel taxa with neosex chromosomes. So I mentioned that Anolis disticus has neosex chromosomes, but a lot of the previous research in neosex chromosomes has come from flies, mammals, and butterflies. So once we have a lot of species with neosex chromosomes, we can kind of put them all together and see what impacts they have and what familiarities they have as well. And lastly, Anolis disticus, I actually never really talked about my model organism as a model organism, but when it's out in the wild, it's a great model for speciation because there are 16 different subspecies that are possibly along a speciation continuum, as well as color evolution. So you can see that there are a lot of different subspecies have very unique colorations, and that could be something that's really interesting to study with gene expression data to understand how different colors are expressed and formed. And with that, I'd like to thank everybody for coming to my talk, and I would love to thank my committee members, my collaborators, and you guys for listening to me. So with that, I can take some questions. Thank you. Hi.
When you looked at TE accumulation, did you parse by TE type, TE family, or anything to look at? Because the idea that like LR, I think LTRs are supposed to accumulate on sex chromosomes, but I don't know if that's been looked at in your species.
That's a great question. So when we look at TE accumulation, I think we just kind of counted all of the ones up that exist. We didn't really parse them by which type they were. We were just looking for their presence. But I can definitely run an analysis that would just be the specific LTRs, and we could see if there's uneven accumulation of those. Great question. Thank you. Anyone else? Last minute. Yeah. Thank you.
So one of the things that has to happen when you have these kinds of fusions like this now, there's two X's, and they have two centromeres, and you have a giant nuclei that only has one. Any idea in your data were you able to parse out what's happening with centromeres in the system?
That's a really good question. We did not, we were not really able to identify our centromeres. Like I said, we kind of had a lot of issues assembling our Y chromosome because it's in two pieces. So I don't know if we actually got the centromeres. But yeah, thank you. All right, it's 10:30. That's my time, everyone. Thank you, guys.
Okay. So I'm Melissa Toups. For 3 more days, I am an assistant professor at Bournemouth University in the UK. And then the 4th day, August 1st, I'll be starting at the University of Louisiana Lafayette. So I'll be talking about some work that looking at the X chromosome in insects. So when we look out in nature, we see that there is a lot of diversity in sex-determining systems. Some organisms, such as the American alligator, uses temperature or an environmental cue to determine sex. Some organisms only have a single gene that determines sex. So for example, the pufferfish, it's literally a single nucleotide polymorphism that is present in males but not in females that determines sex. We're all probably pretty familiar with the X and Y sex chromosomes, where females are the homogametic sex and have 2 X chromosomes, and males are the heterogametic sex and have an X and a Y. But the situation can also be reversed, where the females are the heterogametic sex and have a Z and a W, and males are the homogametic sex and have 2 Z chromosomes, which is what we see in birds and lepidopterans. We also see organisms that have lost the sex-specific chromosome completely, such as several insect species that are X0 or Z0 species. And some organisms go the entire opposite way, as we just heard about, where they go through fusions or reciprocal translocations and add to their sex chromosomes pairs. And all of these are thought to be part of different aspects of or different time periods of a continuum for how sex chromosomes evolve. So sex chromosomes are thought to evolve when a pair of autosomes acquires a sex-determining locus. So in the simplest case of an XY system, you could have a male-determining gene, and if on that same chromosome you have an allele, in this case in blue, that is male beneficial, you would expect selection should favor linkage between the male beneficial allele and the male-determining gene. And the best characterized mechanism we have for this is through an inversion, though that doesn't always happen. And then this leads to recombination suppression, and between the X and the Y chromosome, and lots of changes occur on the Y chromosome, such as degeneration, leading to an accumulation of transposable elements and a loss of genes. This process can repeat itself and eventually result in highly heteromorphic sex chromosomes, like we see in mammals, where we have the very small Y compared to the X, and then this can be lost completely. But of course, nothing is ever that simple, and so there are lots of variations on how this can happen. Different fusions, this process can restart. But what I'm trying to get at is that basically we have all this diversity of outcomes, and we don't necessarily understand what's driving them. So for example, in some organisms, like frogs, sex chromosome turnover is constant, and we see it between frog species and often within a species, whereas other organisms maintain their sex chromosomes for a really long time, such as mammals. And so what this project is really trying to answer this question as to why do some sex chromosomes turn over rapidly, whereas others persist for hundreds of millions of years. And so to do this, we looked at insects to try to understand. So what I'm showing you here is a phylogeny of insects orders and a couple of outgroups. And so just to point out what you're looking at, on the tips of the phylogeny, there are these pie charts, and that tells you what kind of sex chromosome system these different orders have. So in light green and dark green, these are X, Y, and XO systems. In blue are ZW and ZO systems, and then red, for our case, we'll just classify as other. And so a few things I want to stress looking at this phylogeny. These are extremely distantly related taxa. We're talking about roughly 450 million years of evolution on this tree. What you'll also notice is that there's lots of XO and XY systems. And so our question was, is this the maintenance of a single X chromosome or recurrent turnover between XO and XY systems? And some people have been looking at this, I guess I would say over the last probably 15 years or maybe more like 10 years or so, but mostly in like a pairwise manner, looking at what genome they sequenced in their organism and comparing it to what we know about dipterans. And there's been some evidence that it is maintained, that there's homology between the X chromosomes of in certain groups and dipterans. But we wanted to test this in a systematic way. And so what we did is we sampled 10 orders we looked at 10 chromosome level assemblies in species across 10 orders of insects over 450 million years of this evolution. And just to point out a few things about what's going about these species. So lepidopterans are the only ZW system that we sampled. The rest are XO or XY. We also sampled the outgroup to class Insecta, the order Collembola. And we used the blue-tailed damselfly, which is an odonate, as our reference species. And so basically what we did is we wanted to look at which chromosome in our focal species, which is basically everything other than a reference species, shares more genes than you would expect by chance with the damselfly X chromosome. So looking for what is homologous to the damselfly X chromosome. And so in this example, what you see is that in the American grasshopper, it's chromosome X and the X chromosome and chromosome 2 are homologous to the damselfly X. And so we did this for all of these orders, and I'll just march down the phylogeny and tell you what we found. So we didn't have a chromosome level assembly for stick insects, but we did know what was on the X chromosome, and we found that it was homologous to the damselfly X. Within Plecoptera, there were 2 X chromosomes, and both were homologous to the damselfly X. In Lepidopterans, we found that the Z chromosome was not homologous, which has been consistent with other people have found in the past. But then the dipteran ancestral X chromosome, the neuropteran X chromosome, I can't even read this, sorry, the coleopteran X chromosome, the hemipteran, and the Collembola X chromosome were all homologous to the damselfly X. And so we think, and also previous work had shown that the Baltodia X chromosome was also homologous. And so we think that this suggests that it's the same X across all 10 orders, and that this is the ancestral state of insects. And this puts the origin of this X chromosome back to about 450 million years, making it the oldest one known to date. And so then we were curious, okay, so why is the insect X chromosome maintained for so long? And when I spoke about the model before, I talked about how the Y chromosome degrades. And so it loses genes and it accumulates TEs. But the X chromosome also changes too. So we see changes in gene expression to compensate for loss of gene loss in the Y. Genes also flee the X chromosome, and the X chromosome also accumulates TEs, although at a lower rate than the Y chromosome. And so what results is this really highly modified X chromosome that doesn't readily transition back to an autosome. And this is referred to as the evolutionary trap hypothesis. And so if we look within insects, only dipterans have extensive sex chromosome turnover. But when you look within Diptera, dipterans are really nice to look at in this sort of analysis because they have conserved chromosome arms that are known by different letters. So they're molar elements A through F. And what we know, a nice analysis by Beatrice Vicosa and Doris Bachtrag a few years ago showed that the ancestral sex chromosome in dipterans was molar element F. But what they also showed is that if you look at the phylogeny, there's turnover from the F chromosome to literally every other molar element. So what is going on here is just tons of recurrent XY turnover within Diptera. And so the question is, how does Diptera escape this evolutionary trap that seems to have trapped all the other insects as far as we know? And so the one hint we had was that the ancestral Dipteran X chromosome is really small. And so if you look at Diptera compared to the rest, most of the X chromosomes in other insect orders are 1,000 genes or sometimes several thousand genes. But we thought that perhaps the shrinking of the Dipteran X chromosome is what allowed for turnover of sex chromosomes within Diptera, while the ancestral X was maintained in other insect orders. And so to test this hypothesis, we sequenced the genome of the scorpion fly Panorpa cognata, which is a Mecopteran and an outgroup to Diptera. And sort of zooming in on what we did, we wanted to look at, we compared the Panorpa cognata to an outgroup, in this case the migratory locust, as well as two distantly related Dipteran species, the screwworm and the spotted crane fly. And importantly, the spotted crane fly is sister to all other Dipterans. And so we knew once we looked at this that because the spotted crane fly has a small X, that all Dipterans share this really small X chromosome. And so if the shrinking occurred in the lineage leading to Diptera, we expect the Mecopteran X to be large and homologous to the ancestral insect X chromosome, and then also homologous to the Dipteran F chromosome. And so this is what we find when we compare the Panorpa cognata scaffolds to the migratory locust chromosomes, which is what's up here. And so this is a ribbon plot showing, I mean, I guess you guys have seen this now several times, so I don't need to go through too much data, too much explanation, but basically what we find is that the X chromosome of Panorpa, which is scaffolds 1 and 22 in our genome assembly, is homologous to the X chromosome of the migratory locust. And when we compare it to the spotted crane fly, we find exactly what we expect under this hypothesis as well, that the scaffold 1 and scaffold 22 are homologous to the tiny little X chromosome of the spotted crane fly. And so this suggests that the shrinking of the X chromosome did actually occur in Diptera, and that maybe this is why we see this turnover, because this reduced the fitness cost of that transition, of reverting back to an autosome. And so to summarize the talk, the X chromosome is shared across 10 orders of insects in Collembola, which is an outgroup, making it, at 450 million years old, the oldest known sex chromosome. And when we're trying to look at the system to understand why some turn over rapidly, whereas others persist for hundreds of millions of years, it seems that in this example of Dipterans, it looks like they escape the evolutionary trap through shrinking, and therefore we're reducing that fitness cost of transitioning this highly modified X chromosome back into an autosome, whereas other insect orders have maintained their large insect X. And so with that, I will acknowledge the people who I worked with on this project, and I will also say that, reiterate that I am moving to the University of Louisiana Lafayette, and I will be recruiting grad students for fall 2025. And we will be working on neosex chromosomes and hawker dragonflies, and I would love to talk to anyone who's interested about that. And so with that, I'll take any questions. So there's been a sampling by other folks within these orders, right? So there's like within Hemiptera, I can think of a paper that came out several years ago where there are hundreds of millions of years diverge within Hemiptera, and it's the same X chromosome. I think similar work has been done in Coleoptera. I've been working a little bit on odinates, and it looks like it's the same there. I mean, it's of course there's possible that there is turnover in other orders, but we have not detected it on the same scale in Diptera. Now, I granted that the sampling has not been as extensive, but the sampling that we have suggests that it does not look like it's turning over at the same rate. Yes? Yeah. We did not do Hymenoptera. Yeah.
Cool talk. I'm curious about the ZW insect taxa. Do you know if there's
So there's, well, I don't know, I can only speak to Lepidoptera and it's not my work, but I'm pretty sure that there's a recent paper on this that talks about how the Lepidoptera in Z is conserved. I think it's also shared with Trichoptera, but I'm not 100% sure. But I would imagine it is, given how turnover seems to be fairly rare. I would imagine it is. I have no idea what time it is. Okay, well, I'll be around, thank you.